Good evening, everyone. Myself, Roche. Here, we are going to present virus. Introduction. A virus is a microscopic infectious agent that requires a host cell to replicate. It consists of genetic material, DNA or RNA, within a protein coat and sometimes an outer lipid envelope. Viruses infect various life forms, including humans, animals, and plants, and can cause these diseases ranging from the common cold to severe illnesses like COVID-19. They spread through direct contact, airborne droplets, bodily fluids, and contaminated surfaces. Vaccines and antiviral medications are key to prevention and treatment. Morphology of virus. There are morphology of virus. First, capsid. Capsid, protein coat. The capsid is a protective shell made of protein subunits called capsopears. Shape, helical, which is rod-shaped with the capsid forming a spiral around the viral genome. Icosahedral, spherical with 20 triangular faces resembling a polyhedron. Complex, combination of shapes often found in bacteriophages, viruses that infect bacteria which have a head-tail structure. This is the morphology of virus, and this is the structure of capsid, which is present glycoprotein inside genome, then capsid, and outside is coat. There are four types of capsid: helical, polyhedral, spherical, complex. In on helical, that is tobacco mosaic virus, polyhedral adenovirus, spherical influenza virus, complex bacteriophage. Second is envelope. Some viruses have an outer lipid membrane derived from the host cell's membrane called an envelope. Enveloped viruses often have glycoproteins on their surface that help in attaching to host cells. Non-enveloped viruses lack this lipid layer and rely on their capsid for protection and attachment. This is the structure of envelope. Its present envelope protein, then envelope, inside viral genome, then nucleocapsid, then viral tegument. Third is genome. Genome, nucleic acid, viruses can have either DNA or RNA as their genetic material, which can be single-stranded or double-stranded. The genome can be linear or circular and varies in size and complexity. This is the structure of genome. Envelope protein, envelope, viral genome, nucleocapsid, viral tegument. Other structure, which is tegument. Tegument is found in some viruses like herpes viruses. It's a layer between the capsid and envelope that contains proteins involved in viral replication and modulation of the host immune response. This is the structure of tegument, which tegument inside the nucleocapsid is present, and inside the nucleocapsid, dsDNA genome is present, and outside membrane, and this is glycoprotein. Then bacteriophage T4. The T4 bacteriophage is a virus that infects E. coli, Escherichia coli bacteria. That T4 bacteriophage injects its DNA into the bacterial cell, hijacks the cell's machinery to replicate, and eventually causes the cell to burst, releasing new phages. It is commonly used in molecular biology research. Bacteriophage T4 has a distinct structure consisting of head, tail, base plate, tail fibers, collar, and whiskers. Head, that is capsid head, which is icosahedral, contains viral DNA. Tail, which is long and it is contractile shape. Base plate, which is docking structure with tail fibers. Tail fibers aid in attachment to the host. Collar and whiskers connect the head to the tail and provide stability. Then it's adenovirus. Adenovirus are non-enveloped viruses with an icosahedral capsid composed of hexone and penton proteins. The capsid contains linear double-stranded complex with four fiber proteins protrude from the vertices of the capsid and play a role in attachment. Capsid. This is the structure of adenovirus. In the morphology, we will see capsid. Capsid adenoviruses have An icosahedral capsid, which is geometrically regular structure composed of 252 capsomeres, which is protein capsomeres are primarily made up of hexane proteins, which form the majority of the capsid structure, and penton proteins located at the vertices of the icosahedron. Genome. Genome, they contain a linear double-stranded DNA genome, which is non-segmented. Fibers extending from the penton base are fiber proteins that help in attaching the virus to host cells. Size. Adenoviruses are relatively large compared to many other viruses with a diameter of about 70 to 90 nanometers. HIV retrovirus. HIV is a retrovirus that attacks the immune system, specifically CD4 plus T cells, leading to AIDS. Retroviruses insert their RNA into the host DNA, enabling the virus to replicate within the host cells. This is the structure of HIV retrovirus. We'll see the morphology. Envelope outer lipid bilayer with glycoproteins GP120 and GP41 for cell entry. Matrix P17 protein layer providing structural support. Capsid conical core made of P24 protein enclosing the viral RNA and enzymes. Viral RNA, two single-stranded RNA molecules, enzymes reverse transcriptase, RNA to DNA conversion, integrase viral DNA integration into host genome, protease protein processing for new virions, accessory proteins, regulatory proteins aiding in replication and immune invasion. If we will see in HIV retrovirus, it's present inside RNA. Outside the RNA, its capsid, then reverse transcriptase inside the RNA, then matrix proteins, viral envelope, and these are glycoproteins.
Hello, good evening. My topic is types of viruses. There are two types of viruses, RNA and DNA viruses. First, we go to RNA virus structure. RNA virus can have either single stranded and double stranded RNA genome. They are basically enclosed within a protein capsid or nucleic acid. Types of RNA virus. There are three types of RNA virus. Positive sense RNA virus, negative sense RNA virus, double stranded virus. First, positive sense RNA virus. There are RNA can be directly translated into proteins by host cells. Example include coronavirus. Examples include coronavirus, hepatitis C virus, and Zika virus. Positive sense RNA virus. अरे इसको बंद कर हेलो positive sense RNA virus. They can be directly transmitted into proteins by host cell ribosome. Example include coronavirus, hepatitis C virus, and Zika virus. Negative sense RNA virus. There are RNA must be transcribed into a positive sense RNA before protein synthesis. Example include influenza virus and Ebola virus. Double-stranded RNA virus. These viruses have RNA genome that are double-stranded. They include viruses like retrovirus, which cause gastrointestinal infections. Here is the type of RNA virus. There are two types like single-stranded RNA and double-stranded RNA. And there are other two types in single-stranded RNA. There are positive sense strand and negative sense strand. In positive sense strand, there are two types, non-enveloped and enveloped. In negative strand, there is only one enveloped. In double stranded, there is only non-enveloped. It includes Reobacteria, which can cause gastrointestinal infection. Now the second type is DNA virus. Structure DNA virus have double-stranded DNA genome enclosed within a protein capsule. Some may also have outer lipid envelope derived from the host cell membrane. Example, herpes virus, adenovirus, which can cause a gastrointestinal infection and respiratory infections, and poxovirus, which can cause smallpox virus. Here we can see the DNA virus. There are two types, double stranded and single stranded. In double stranded, there are enveloped and non-enveloped, which includes adenoviridae, papilloma viridae. In enveloped include poxoviridae and herpesoviridae. In single stranded DNA, there is only non-enveloped, which include paroviridae. Some of the examples of virus like coronavirus, we recently gone through this virus, coronavirus, which is derived from SARS-CoV-2. Coronavirus are named for their crown-like appearance due to the spike protein that protrude from their surface. Structurally, they are enveloped virus with a positive single-strand RNA genome. The viral practice consists of four main structural protein: spike protein, envelope protein, membrane protein, nucleocapsid protein. Spike protein responsible for binding the host to cell receptors. Envelope protein plays a very important role in resembling and release. Membrane protein helps in the shape of viral envelope. Nucleocapsid protein bind to the viral RNA genome. We can see here the structure. We have spike glycoprotein, membrane protein, and nucleoprotein, genomic RNA inside envelope small membrane protein. Genome. It consists of single standard RNA genome that encodes several structural and non-structural protein essential for viral replication and infection. Transmission. The primary mode of transmission is through respiratory droplet from infected individual during coughing, sneezing, or talking. It can also spread via aerosol and contact with the contaminated surface. Pathogenesis. SARS-CoV-2 primarily targets cells lining the respiratory tract, particularly those expresses the ACE2 respirator. This respirator binding facilitates viral entry into host cells, leading to respiratory symptoms ranging from mild to severe pneumonia. Clinical manifestations. COVID-19, the disease caused by SARS-CoV-2, presents within the symptoms such as fever, cough, shortness of breathing, fatigue, and loss of taste or smell. Severe cases can lead to acute respiratory uh, distress syndrome to multi-organ failure and can lead to death also. Prevention and treatment. Prevention measures include vaccination, wearing mask, and hand hygiene and physical distancing. Treatment focus on supportive care, antiviral medication, and in severe cases, oxygen therapy and ventilatory support. Now, second example is influenza virus, which belongs to the family called Orthomoxiviridae. Influenza, it has a characteristic structure. It is enveloped and spherical in shape with a diameter of about 80 to 120 nanometers. The viral envelope is derived from the host cell membrane and consists of two main surface, glycoprotein, hemagglutinin, and neuraminidase. This protein play a crucial role in viral attachment to the host cell and in release of a new virus particles from infected cells, respectively. Inside the envelope, the virus has a matrix protein layer surrounding its segment, negative sense RNA genome. We can see the structure here. Outer layer, we have hemagglutinin and neuraminidase. And inside, we have nucleoprotein genome. The genome of influenza virus consists of 8 RNA segments that encode essentially protein, including hemagglutinin and neuraminidase, which are critical of viral entry and release. Transmission similar to SARS-CoV-2, influenza spread through respiratory droplet aerosol and contact with the contaminated surface. It causes seasonal epidemics and occasional pandemics. Pathogenesis. Influenza virus primarily infect the respiratory epithelium, causing inflammation and damage. The virus binds to sialic acid respiratory on the host cells, initiating infection and replication. Clinical manifestations. Influenza typically present with the sudden onset of fever, chill, headache, muscle ache, cough, sore throat, and fatigue. Complication can include pneumonia, exacerbation of underlying conditions, and in severe cases like death also. Prevention and treatment. Prevention strategies include annual vaccination, respiratory hygiene, and antiviral medication. Example, oselmet for treatment, especially when administered early in the course of illness. Now, virus contribute to disease through several mechanisms. First, infection and replication. Infection and replication. Entry. Virus attach to and enter the host cells using specific receptors. Replication. Once inside, they hijack the host cells machinery to replicate their genome and produce new viral particles. Cell death and damage. Cytopathic effect. Viral replication can cause a direct damage to the host cells, leading to the cell death. Apoptosis. Some viruses include programmed cell death, apoptosis to evade immune response. Immune response evasion. Immune response suppression. Virus like HIV weaken the immune system, making the host more susceptible to the other infections. Antigenic variation. Some viruses like influenza change their surface protein to evade immune detection. Fourth, chronic infection. Recurrent infection. Some virus like herpes virus can remain in the host reactive later, causing recurrent disease. Persistent infection. Virus like hepatitis B and C can cause a long-term infection leading to chronic disease and complication, triggering immune responses. Inflammation. Viral infection can cause excessive inflammation, contributing to tissue damage. Autoimmunity is viral infection can trigger autoimmune disease, whereas the immune system attacks their own body tissue. Oncogenesis. Some viruses like human papilloma virus and Epstein-Barr virus can lead to cancer by causing genetic mutation and altering cell growth regulation. Through these mechanisms, viruses can cause a wide range of disease from acute infection like a common cold flu to chronic conditions and cancer. Thank you.
Thank, thank you so much. Thank you, ma'am.